Welcome to our seminar series, direct to you from where it is happening. This is the first of the series, which I hope you will enjoy. I'm Kazu Igarashi from Tohoku University in Japan. Today, I'd like to bring you through our research on ferroptosis, iron-dependent cell death. After a brief introduction of ferroptosis, I will describe how the transcription factor BACH1 promotes ferroptosis. Then I will try to convince you why the regulation of ferroptosis by BACH1 is important to understand disease processes with future translation possibility to new therapeutics. Toward the end of my talk, I'd like to share with you our recent research theme, which is propagation or spreading of ferroptosis among cells. So let us begin. Here is a poem by an English poet, John Donne. Why grasses are green or why our blood is red are mysteries which none have reached unto. This poem testifies the curiosity of the human beings, which has driven so many scientists to unravel the component responsible or essential for the colors, which is iron. In cells, iron is used to generate iron-sulfur cluster, or heme. These molecules bind to various proteins to function in respiration, photosynthesis, enzymatic reactions, and oxygen binding. All of these reactions are essential for cells and organisms to survive. On the downside, iron can be very toxic to cells. Ferrous iron can catalyze the conversion of molecular oxygen to reactive oxygen species, such as hydroxyl radical. Among the enzymes in the human body, 6.5% are iron dependent. Among those, a half is using iron as heme and 17% as iron-sulfur cluster. As such, iron is essential for most of the life forms. Of course, there are always exceptions, like some residue of microbes. We have around 3 to 4 grams of iron in our body. The majority of the iron is present as a component of hemoglobin. As I mentioned, there are many other proteins containing iron, reflecting its very high metabolic activities. The liver contains 1 gram of iron. However, the highest demand for iron is for making new red blood cells in the bone marrow. Roughly 20 mg of iron is used every day for generating red blood cells afresh. This amount of iron is basically supplied by reusing iron in the red blood cells. Macrophages phagocytose senescent red blood cells, breaking down hemoglobin, and heme oxygenase 1 scavenges iron from heme, releasing iron into the blood. Of course, our body loses iron via dying cells in the gut or as scarf of the skin. The loss of iron is replenished by daily intake from food. Such metabolism, dynamics, and homeostasis of iron in our body is regulated by many protein regulators, such as transferrin, its receptor, ferritin, ferroportin, and heme oxygenase 1. Ferroptosis is cell death due to lipid peroxidation in the cell membrane. This lipid peroxidation is catalyzed by ferrous iron and counteracted by the reducing activity of glutathione. When ferrous iron is increased and glutathione is decreased, or when the balance of these two elements is skewed, ferroptosis can take place. We have found that BACH1 promotes ferroptosis by increasing iron and by decreasing glutathione. How? BACH1 replaces the expression of genes for glutathione synthesis, including cysteine transporter and enzymes for its synthesis. Another set of BACH1 target genes is related to iron. including ferritin and ferroportin. By altering the metabolism of two key elements of ferroptosis, BACH1 promotes this type of cell death. Studies of ferroptosis using various disease models have suggested its involvement in disease processes, including renal diseases, cardiac and brain infarction, steatohepatitis, and infections including COVID-19. Alzheimer's disease and other neurodegenerative diseases have also been suggested to involve ferroptosis. In these diseases, ferroptosis contributes to tissue damage. In contrast, in the case of various cancers, ferroptosis appears to function as a protection or tumor-suppressor mechanism by killing cancer cells or precancerous cells. We have been working on BACH1 and its closely related factor BACH2. BACH2 expression is very specific to cells like B and T lymphoid cells. In B cells, it is required for proper activation of B cells in response to antigen, including antibody class switch recombination and somatic hypermutation of antibody genes. In contrast, BACH1 is widely expressed in many types of cells and tissues where it regulates the expression of genes related to iron, including globin genes. Our research on BACH stemmed from our studies on the regulation of globin genes by NFE2, which has three other related factors. For example, NRF2 activates oxidative stress response. The upper four factors function as activators of gene expression. In contrast, BACH1 and BACH2 repress their target gene expression. Personally, I have been obsessed with these factors because of this single finding. When we purified recombinant BACH1 protein from E. coli, the solution was brown-red like this. This was a big mystery for us. Finally, it turned out that it is because of direct binding of heme to the protein. Heme inactivates BACH1 or BACH2 by inhibiting its DNA binding activity, inducing nuclear export, and increasing polyubiquitination and degradation. Therefore, metabolic activities, more specifically heme, can induce the expression of their target genes by inactivating BACH1 or BACH2, meaning that these factors organize or orchestrate circuits of metabolism and genes. To understand the roles of such circuits in diseases or immune response is what I think is very interesting and important. Okay, now let's examine closely the role of BACH1 in ferroptosis. Here, we isolated fibroblasts from wild-type or BACH1 knockout mice and treated them with elastin, an inducer of ferroptosis. Elastin is an inhibitor of the cysteine transporter. The wild-type cells underwent ferroptosis, whereas the BACH1 knockout cells were more resistant to the drug. Importantly, necroptosis or apoptosis was similarly taking place in these cells. By RNA expression profiling, the expression of ferritin genes and ferroportin was higher in the knockout cells than in the wild-type cells. Upon mapping of BACH1 binding sites across the genome, these genes showed direct binding of BACH1. These results suggest that BACH1 directly represses the expression of iron-related genes. So what is happening to iron in these cells? Here, we measured cellular ferrous iron using fluorescent indicator and cell sorter. Under the normal conditions, BACH1 knockout cells showed higher level of iron. This is something we do not understand, but please see this. When ferroptosis is induced with elastin, iron was increased in the wild-type cells. In contrast, the increase was much less in the knockout cells. The increase in the wild-type cells appears to involve ferritinophagy, reducing ferrous iron from ferritin. We also found that glutathione is lower in BACH1 knockout cells. Therefore, BACH1 promotes ferroptosis by increasing iron and decreasing glutathione. The next question is whether BACH1 regulated ferroptosis is involved in diseases. The first example I'd like to share with you is Parkinson's disease. The second is anemia. This is an exciting collaboration with Professor Bobby Thomas of the South Carolina Medical School, United States. Professor Thomas and his colleagues examined BACH1 protein amount in the posthumous brains of healthy or those who had suffered Parkinson's disease. BACH1 protein was higher in the diseased brains. Then they asked whether BACH1 inhibition would alter disease phenotype in mice. Here, by treating the mice with a drug called MPTP, death of dopamine neuron was induced. This is one of the widely used models of Parkinson's disease. By the way, the story of MPTP is very interesting and you should check it out later. Anyway, with MPTP, dopamine neurons were reduced. When the mice were treated with a new inhibitor of BACH1, HPPE, the loss of cells became less. At this moment, we do not know if the cell death in response to MPTP is ferroptosis or not. Nonetheless, BACH1 seems an interesting therapeutic target. Next example is anemia. The synthesis of heme starts from condensation of succinyl-CoA and glycine in the mitochondria, which is catalyzed by ubiquitous enzyme ALAS1 or its erythroid-specific isozyme ALAS2. The product of this reaction, delta-aminolevulinic acid, undergoes several enzymatic reactions in the cytoplasm and additional reactions back in the mitochondria, generating protoporphyrin. Finally, iron is inserted into the protoporphyrin ring. Some of anemia is caused by mutations in ALAS2. These mutations cause a reduction in heme in the progenitor cells of red blood cells, leading to a reduction of hemoglobin, and hence anemia. Because iron is not used in those cells, iron is accumulated within mitochondria, leading to the formation of typical ring sideroblasts in the bone marrow. This project is a collaboration with Professor Harigae and his team, Department of Hematology at Tohoku University. They found mutations of ALAS2 of their patients suffering from sideroblastic anemia. To investigate how these mutations would affect erythroid cells, they generated human hematopoietic stem cell lines with those mutations using gene editing. Then they induced differentiation of those cells to red blood cells. In these cells, BACH1 protein was increased compared with the control cells, whereas one of the protein for glutathione synthesis was reduced. This was reflected in the reduction of glutathione. Furthermore, when these cells were treated with elastin, they showed more cell death compared with the control cells without the gene editing. In one of the cells, the increased cell death was rescued by adding iron chelator. These results suggest that the ALAS2 mutations not only cause a reduction in hemoglobin, but also increase ferroptosis in the hematopoietic cell fraction. Now a new enigma in ferroptosis. We found that ferroptosis can spread or propagate from cells to cells. What did we observe? Here is one of the experiments. We treated fibroblastic cells with elastin. After 24 hours, there are many cells still alive but destined to die. Those cells were washed to remove elastin, fed with new media without elastin, and then co-cultured with another batch of cells marked with orange fluorescent protein. Cell death is monitored with annexin 5 in green here. In half or 1 day, these orange cells also started to die. With this and other experiments, we came to conclude that ferroptosis can propagate from cells to cells, and the propagation is mediated by some materials secreted from the dying cells. One of the limitations of ferroptosis research is the use of drugs to induce ferroptosis, like inhibitors of cysteine transporter or GPX4. As a genetic model, tissue-specific knockout of GPX4 is also used. Each model is very powerful and has driven so many wonderful findings, but they have also limitations. By extending our findings, we have established a new model cell system of ferroptosis. In this system, we used BACH1 knockout fibroblasts and re-expressed BACH1. Upon BACH1 expression, these cells underwent rapid ferroptosis. It is so great to be able to induce ferroptosis without using drugs. This system is ideal to investigate the mechanism and biological significance of ferroptosis propagation. It may also be used to identify biomarkers of ferroptosis. Okay, I'd like to finish up this lecture by introducing and thanking my collaborators. Dr. Nishizawa has been leading our project on ferroptosis for over 8 years. Without him, we would have never been able to start working on and to venture into ferroptosis. And we are collaborating with many wonderful teams shown here. That's that. Thank you for joining me, and goodbye until next time.